Okay, so welcome to today's webinar. Today we'll be giving you an introduction to FCS Express image cytometry. And I'd just like to take this opportunity to introduce myself. My name's Kaya Ghosh. I'm a technical application specialist with Novo software. And Novo software has been around for about 15 years. We've been making analysis software for flow cytometry data. And in 2011, we launched this FCS Express image cytometry product, which you can use to analyze your image cytometry and microscopy data. Okay, so whether you're new to the world of image cytometry or you've already started analyzing your data sets using image cytometry, you've probably noticed that image cytometry shares a lot in common with flow cytometry in terms of data analysis. In other words, they both involve taking multi-parametric data sets, be that multi-channel fluorescence data or multiple morphological features that you're collecting from your cells or other objects, and analyzing that data by plotting it in 1D plots like histograms or 2D dot plots or contour plots, and looking for statistically different changes among the different populations that you determine based on gates that you set subjectively. Okay, and so what FCS Express image cytometry allows you to do is to leverage those powerful analytical tools that already exist for flow cytometry to analyze your multi-parametric data sets and then to relate that analysis back to the original images for confirmation. And so rather than doing that on a one-by-one basis where you're wading through a backlog of images or maybe looking through very complicated Excel sheets for that needle in a haystack, what FCS Express image cytometry will allow you to do is to spend less time going through all those data sets manually and rather analyze those images in bulk more efficiently, which also facilitates making conclusions in a more objective manner. Okay, so that's an overview of what we aim to do with our product. And so what we're going to do today is we're going to do a little bit of an analysis together. We're going to build a layout together and then look at a couple of example layouts so you can see a little bit about what the product can do. And as I go along, I just want to reiterate that if you do have a question, just enter it into the GoToWebinar control panel here at left where it says enter a question for staff. And either the staff members that we have on the line or I'll get back to you during the course of the webinar via that chat window. Okay? Great. So let's talk a little bit about the workflow for analyzing image cytometry data. As you can see in this schematic, the workflow consists of two phases. The first phase is image data generation, and the second phase is data analysis and report generation, which can be easily accomplished in FCS Express image cytometry. So let's talk a little bit more in detail about the first phase. The first phase is image data generation, and the first step there, of course, is image acquisition. And so you can acquire your images in a number of ways. You can acquire them using a traditional microscope. You can acquire them using an image cytometer. And I just want to note at this point that FCS Express is compatible with a number of different manufacturer formats that are out there. So if you're using equipment from Molecular Devices, Fluxion, Nexalom, the Life Technologies Tally, Chemometic, Perkin-Elmer, those are just a few of the manufacturer formats that we're compatible with. So if you're acquiring your images on those hardware, we're compatible with those. And even if you're not using one of those pieces of equipment, any image that you acquire, even if it's off of a very traditional microscope with a cheap digital camera, we have pathways to compatibility that will allow you to analyze those images in FCS Express. So you can use freeware such as Cell Profiler or ImageJ to basically segment those images so that you can analyze them in FCS Express. Okay, and so that brings us to the second step in image data generation, and that's image segmentation, okay, which can be accomplished by Cell Profiler, ImageJ, or many of the softwares that come along with those manufacturers that I just mentioned. And so image segmentation in a nutshell means if you have, let's say, a field of view where you're looking at a plane of cells. You have, let's say, 100 cells in your field of view. Image segmentation aims to segment that image into 100 different objects, where one object is one cell, using the most simple example. Okay, so that's what the software or the freeware will allow you to do. And then the next step is feature extraction, which is also performed by that software or freeware that you're using, which will allow you to distinguish different features among the different objects. So let's say cell shape, cell size, intensity of fluorescence for that given object. Okay, so once that whole first phase is accomplished, then you progress to the second phase, which is data analysis and report generation and communication and publication, which is very easily accomplished in FCS Express. Okay, so for those of you who are familiar with flow cytometry or who have already begun to work with image cytometry data, you know that the data analysis is very similar to flow cytometry, where you're making a number of 1D or 2D plots, creating gates or markers around populations of interest, and then getting summary statistics based off of those populations. Okay, and that's all done very efficiently in FCS Express, which I'll show you. And it's very easy to get those summary statistics off of large data sets. But in addition to that capability, you can generate very nice reports in FCS Express, and that's because we have advanced compatibility with Microsoft Office applications such as PowerPoint and Word and the like. So we have a very seamless transition between those things. And we can even generate PDFs. And we also have a number of communication and publication tools which enable you to collaborate with your peers very easily. And I'll talk a little bit about that at the end. Okay, so that's just an overview of the workflow there. So what I'd like to do with all of you now is let's get started and build a very simple image cytometry layout together. All right, so what I'm going to do is just start by creating a new layout. All right, and so before we actually start adding data to this layout, you can see that the appearance of FCS Express very closely resembles Microsoft Office PowerPoint or Microsoft Office Word, and that's very much on purpose. We've designed the software to mimic that interface so that it'll feel familiar to you right from the start. So if you already spend time dealing with ribbons and tabs, like you have in Microsoft Office PowerPoint, for example, you'll feel right at home in FCS Express. So this ribbon and tabs interface arranges buttons into logical groups so you can easily find the function you're trying to do. So here on the Home tab, we have a number of frequently used functions, such as adding a new page. We have a number of different layouts. Just like in PowerPoint, you have a number of different slide templates you can use. We also have the gating functions here on the Home tab, since those are so frequently used. If I come over here to the gating tab, you'll see we have a whole bunch of other gating functions as well. The Insert tab allows you to insert different plot types as well as text boxes, okay, in much the same way you'd insert a JPEG image or a text box into PowerPoint. Okay, so it's very easy to get used to, I think. So let's go ahead and get started. Okay, I'm going to start out by adding a picture plot. And you probably don't really know what a picture plot is at this point, but just bear with me here. I think it'll become clear. So I just click once on the layout. I'm immediately prompted to navigate to my data. So let's go to where our data lives. Okay, so what I'm going to do now is add one of these .cpout files. Okay, and so these files are outputs from Cell Profiler. They're based off of TIFFs that were run through Cell Profiler and segmented and had the features extracted. Okay, so this is a very simple data set. I'm just going to go ahead and add that first cpout file. Okay, so you can see hopefully that if I zoom in here, you can see that this is a microscopic field with a number of cells, and it looks a lot like your original TIFF image or JPEG image. But what's different about this picture plot is all of these individual cells have been segmented according to the segmentation routine that was defined in Cell Profiler. Okay? So let me zoom back out, and now I'm going to add in the nuclear picture plot that corresponds with this field of view. And I'm going to do that in a slightly different way. Let's open up our data list. And what I want to show you now is that we have quite a lot of drag and drop incorporated into FCS Express. So what I'm going to do to add the corresponding nuclear cell profiler picture plot is just click on it from the data list and drag and drop it over onto my layout. Okay, so now I have my nuclear image. Let's change the axis here to be looking at the DNA. Okay, and so since these are both objects in FCS Express, I can easily resize them and move them around. You might have noticed. I can even choose to align them to make my layout look a little more neat by coming up to the Home tab and aligning. Okay? So it's really easy to manipulate your objects in FCS Express. So what's the first thing you would do if you were dealing with flow cytometry data? You would probably start creating a gate on populations of interest, right? So let's do the same thing now with our picture plots. I'm going to make an ellipse gate and draw it on the central cluster of cells in my picture plot. And let's call this our picture plot gate. All right, so you can see we have this picture plot gate here, and I can move it around, resize it at will. I have the number of objects, or in this case cells, that fall into my picture plot gate. Okay, I can do the same thing and even create a gate here on the nuclear image. Let's do that. And this time I think I'm going to create it around this group of dim cells over here. Okay, and so at this point I can even insert a gate view to look at a legend of the gates that I have so far. If I change my mind and I want to rename one of these gates, you know, let's do that so the names are more in parallel. So this was my nuclear picture plot gate. Okay, let's call this one our cytoplasmic picture plot gate. All I have to do is double click on it on the gate view and rename it. Okay, so it's very flexible. It is very easy to amend your layout as you go. So let's now create some traditional dot plots or color dot plots so we can start to look at the characteristics of these populations in a little more detail. So I'm just going to come up to my Insert tab and click on Color Dot Plot. Okay, and let's draw it over here. I'm going to zoom out a little bit. And let's also create a corresponding color dot plot from the nuclear image. Notice I'm drawing it to the size that I want. Okay, so when you look at these two dot plots, it might be hard for you to see the dots. They're a little bit small by default, which might be appropriate if you're looking at a field of view with thousands and thousands of objects. But in our case, we probably only got a few hundred cells here. So what I'm going to do is increase the dot size so it's easier for us to see in the webinar. And that's very easy to do. You can just drag around the objects to select them. I'm going to come up to my Format tab, and let's increase the dot size here to let's say 6. We'll make it pretty big. All right, so I think hopefully you can all see those dots pretty well. Maybe I'll make it a little bit bigger even, so those of you with small screens won't have so much trouble. Okay, so you can see that these dots are now colored according to where they fall in the gates. So we have some black dots, we have some red dots, we have some blue dots. Okay, so what I'm going to do now is change the parameters so we can start to look at some of the features that were extracted by Cell Profiler. And let's leave the x-axis here as area of the object, and I'm just going to go ahead and change the y-axis to, let's say, intensity of the cytoplasm. Okay, and I'm going to change this right plot here similarly to intensity of the nuclei. Okay, so we can actually apply these gates that we've created on our picture plot to these dot plots simply by dragging and dropping. So if I only want to look at my cytoplasmic picture plot gate on this dot plot, all I need to do is drag and drop it, and now you can see we're only looking at the red cells. Okay? So remember, if there's any questions as I go along, just go ahead and enter them into the questions and answer box, and I'll be happy to answer them. Our staff members on the line will answer them. Okay? Another nice thing you can do is you can ID outliers. So if I wanted to maybe only look at, well, let's do this on the nuclear plot. Let's say we only want to look at this very, very bright nucleus right here. I'm just going to move my gate over and resize it so it only contains that one cell, that one nucleus rather, and now I can drag and drop it. In this case, I want to do it from the gate view, and I can see where that dot lies on my dot plot. So we see that it has an area of about 500-ish and an intensity of about 0.3. Okay, so it's really easy to do that as well. So, whoops. Okay, and let's just ungate that. So once you start creating gates, you can also create gates here on your dot plots as well, and conversely apply them to the picture plots. So for instance, maybe we only want to look at our very small bright cells here on the dot plot. So I'm going to create a rectangular gate here around this cluster up here, which is relatively small and relatively bright. Okay, and maybe I'll move this text box down so we can see it a little bit more clearly. And so now you can see on the picture plot that that purple color has been automatically backgated onto the picture plot, and we can see where those cells lie. Okay, and so I can continue to adjust that gate, and I can drag this gate to the picture plot to only look at those cells. Okay. In addition, you can change your gating hierarchy. So right now we have all of our gates at the same level in the hierarchy. But if I change my mind and I want my small bright gate to be a child gate of my cytoplasmic picture plot gate, I can just drag it in my gate view to be a child of that gate. And so now you see only cells that fall inside the cytoplasmic picture plot gate are defined as small and bright. So that purple backgating is restricted to this red picture plot gate. Okay, so just like you can change your gating hierarchy for flow cytometry data at any time in FCS Express, you can do the same thing with your image cytometry data. Okay, so once you've created some gates, you probably want to get some summary statistics, and so that's really easy to do. Just right-click on any plot and come over here to Gate Statistics, and now I have a very nice summary table, which itself can be formatted again by right-clicking. So hopefully you've come to see that anytime you want to reformat an object in FCS Express, just try right-clicking on it. So let's right-click here and get rid of some of these columns that we're not interested in. And maybe I'm going to add in some of the columns I am interested in, like X and Y standard dev. Okay? And I could do the same thing here on the nuclear dot plot, of course. All right, so let's save this layout real quick. And let's save this to our desktop. And now I just want to show you how easy it is to transfer your analysis to PowerPoint or PDF or what have you. Okay, so let's save this real quick as a PowerPoint. I'm just defining the path here. And this should open automatically. Okay, so here's our PowerPoint file with our analysis, and you can see that it looks just like our analysis workspace in FCS Express. So there's no guesswork. Your layout in FCS Express is going to transfer very faithfully to PowerPoint or PDF or whatever you choose for your output. And what's more, all of these are transferred over as independent objects, so I can move them around, I can resize them at will. Okay, and they're transferred with very high resolution, so you never need to worry about resolution options in FCS Express. So you can see that it's really easy to create posters and presentations and the like. I can even use my clipboard and transfer over images from different layouts into the same PowerPoint file. Okay, just by doing Control-V and Control-C or using your mouse. So now you see if you have to make a poster or presentation, FCS Express really takes the pain out of that. Okay, so let's go back to our layout here. There's one thing I didn't show you, and that is data grids. So you have a number of different objects here, and it's possible to look at those in a gallery view in FCS Express. So you can look at them cell by cell if you're interested in. So it's real easy just to highlight the object of interest and then come over here to your Insert tab and click on Data Grid. All right, so now you can see if I scroll down that we have about 300 objects that were discerned in this image by Cell Profiler, and we have this table here with the different morphological features that were extracted by Cell Profiler as well. Okay, and what I'm going to do is right-click on this and get rid of some of the parameters that we're not interested in looking at right now. So let's choose only to look at the area and maybe the perimeter. And let's also look at the intensity. Okay, and we're also going to add in these thumbnail pictures. Okay, so let's expand this out so we can see. And this is a data grid which could be gated on the gates you've created, just like you gate dot plots. So if I only want to look at my cytoplasmic picture plot gate, I'll just drag it over to my data grid and see that there's only these different cells that are in it. Similarly, I can even drag over this nuclear picture plot gate, which only contains the one object, and just look at that particular cell of interest. And if you want to zoom into those thumbnail images, all you need to do is expand the row size, and then you can see the thumbnail images blown up in the gallery view. Okay? Are there any questions about that so far? Okay, great. The other thing I want to show you is basically it's very easy to scroll through all your data in FCS Express. So you might recall if you look at our data list here, we've got 4 images, and right now I'm only looking at 2 of those picture plots on my layout. And these are called cells 05 CP out, so they're from a 5-minute time point. But we also have 10-minute time points in this data set, and so if I want to look at those, all I need to do is click Next, and all of the objects on my layout advance to the next data file in the data list. Okay, so now we're looking at those 10-minute time points. And if you had a much larger data set with, let's say, 50 or 100 or more images, and you wanted to have a nice summary of your whole experiment, the best way to do that is with a batch process. And so I'm going to show you real quick how that works in FCS Express. Let's call up our batch actions. Okay, so the first thing I'm going to do is let's do a save to PowerPoint, and let's send this to our desktop again. And let's also add in a print action. Okay, and you can choose between all or just one of the pages if you want. And we're also going to add in an Excel export. Okay, and let's just define the file path here again and the file name. Okay, So we just need to tell FCS Express which statistics that we'd like to export to Excel, because as you can imagine, we have 20 or 30 different parameters for every object, and we don't want to look at a huge Excel sheet with all those parameters that we can't even begin to make sense of. So it's much better to drag over only the statistics you're interested in. So again, we have a lot of drag and drop in FCS Express image cytometry. So if I want a statistic that's based on my red gate here, I'm just going to drag my gate over and drop it into the Excel action. And now I just need to tell the software which statistic is it that I'd like to export. So let's choose percent of gated cells, and we'll just add a title here so it shows up in our column titles in Excel. Okay. You can even drag over your whole plot and choose maybe keyword, well, not keyword tokens because there's no metadata with image cytometry data, but you can choose other statistic tokens. So in this case, I'd like to choose the FCS file name. Okay, so that way we'll know what we're looking at in the Excel spreadsheet. And I'm going to actually reorder my Excel exports because I want that to be my first export, my first column in my export. Okay, so before I click the lightning bolt run to run my batch actions and get my exports, let's just take a look at these checkboxes. So anytime you have a queue lined up in your layout, you can choose whether or not to include each of those actions. So I'm not really hooked up to a printer right now, so I'm going to uncheck that print. But if you were hooked up to a printer, you could just print out the layout as is, and it'll come out just how it looks on the screen on your printer page. Okay, so let's click Run and see what we've got here. Okay, so our Excel export popped up first, so let's just take a quick look at that. Okay, so we have two different file names that are here. And we also have the statistics that popped up. So if we had dragged over gates from the nuclear images, that information would be here too in different columns. But this is a very simple export where we only chose a couple of stats to look at. All right. And now let's take a look at our PowerPoint, which I think is over here. All right, so we have two different slides because we had two iterations in our data set. So we had a 5-minute time point and a 10-minute time point. And if I zoom in here, you can see that, again, these were exported as individual objects and they're very high resolution. And if you come down here to the next slide, this is our 10-minute time points, and you can see that those gate statistics have updated accordingly. And there's even a way to adjust the gates between different samples in your data list. So for instance, in this 10-minute time point image, if I wanted to make this picture plot gate smaller or maybe add different parameters to my data grid or whatever it is, you can do that by adding a pause in your batch actions. So I'm not going to be showing that to you today, but we do have a webinar all about batch processing. And if you have questions about how to add a pause to your batch actions so you can adjust your gates between different samples, just don't hesitate to let us know and we can even help you out later on after the webinar. Okay, so that is building a very simple layout in a nutshell. What I'd like to do now is just show you another example or two of what FCS Express can do with your image cytometry data. Okay, so let's take a look at this next layout. Okay, hang on. I think it might have accidentally gotten closed. Just a sec. Okay, here we go. So this is our nuclear translocation assay, and you can see that this is a 96 well plate, so we could read in 96 well plate data from a number of different manufacturers. And if you take a look at this schematic here, this is basically a schematic of the experimental scheme where a number of different drug concentrations and a number of different drugs were tested for their ability to induce translocation of protein from the cytoplasm of the cells to the nucleus. Okay, so let's take a little bit of a look at what this data really looks like. I'm going to insert a picture plot. Let's make it nice and big so you can see it. And you know what, let's even get rid of some of this color because we have all this automatic backgating here onto our picture plot from the gates that were previously defined in our layout. But I think that's a little bit distracting, so let's do this. Okay, so you can see from our gate view we have all these different gates that were introduced into our layout, and I'll talk a little bit more about what all of those mean later on. But just to show you, we can gate this picture plot. Currently it's not gated, but let's gate it on our single well gate, okay? And let's just see which well that's on. And I'm just right-clicking, coming to Edit Gate, and you can see that the single well is here on this well. But let's move that single well gate onto one of our negative controls, okay? So now I'm gated on well A1, and this is my negative control well where you can see the protein is still in the cytoplasm of the cells. That's why the cytoplasms are all lit up and the nuclei are comparatively dark. All right? If I change my mind and want to make that single well gate be on one of my positive control wells, you can see that the nuclei are lit up while the cytoplasms are dim. Okay? And note that FCS Express can look at your data whether you are looking at RGB images or your grayscale images. In most cases, people choose to analyze grayscale images, even if you acquire an RGB, but it's up to you. I just wanted to show you that we have the capability to do both. Okay, so the purpose of this experiment, again, was to look at the ability of these different drugs and different drug concentrations to affect translocation of the fluorescently labeled protein from the cytoplasm into the nucleus. All right, so let's take a little bit of a closer look at the data by looking at these dot plots. So at the top we have all the cells, and on the x-axis we're looking at the fluorescence intensity at the edge of the cytoplasm versus on the y-axis the fluorescence intensity in the entire cytoplasm. So if you think about translocation occurring in the cell, as the protein migrates from the cytoplasm to the nucleus, the intensity of the fluorescence at the edge of the cell is going to decrease, right? So that's why once we look at our positive control well, which is the translocated events, you can see that there's much less fluorescence intensity at the edge of the cytoplasm, okay? Compared to the negative controls where you have a little bit more of a heterogeneous population. So you've got some cells that do have measurable intensity at the edge of the cytoplasm, and you also have cells that are scattered further left on the x-axis. All right? So it's hard to look at this data and just make sense of it subjectively, and that's where drawing gates on your multiparametric data really comes into play. So on our next page here, we have our translocated cells, or our positive controls, next to our negative control picture plots here. All right? And so looking at these positive controls, we've drawn this blue translocated cells gate here around the population. So since translocation has occurred in these cells, we're drawing the gate just outside of that last cell so that all of the translocated cells are contained within that gate. Okay? And then we've drawn a gate around the rest of the population, or around the rest of the space on this dot plot to include the non-translocated cells. And if you come over here and look at your non-translocated control, you can see that you've got more than 50% of your cells falling into the non-translocated gate, but you've also got a little bit less than half of the cells falling into your translocated gate. So you might not be able to tell by eye, just looking at this image, that you've got a heterogeneous cellular population in your negative controls. But that's what drawing the gates on your image cytometry data really allows you to do. It allows you to define your populations a little bit more objectively than you would be able to do just by eye. Okay? So let's take a look at what we did with that data in the next step. Okay, so just like I showed you in the previous layout that we built together with the cell profiler data, you can create gates on your picture plots or gates on your dot plots and have them automatically gated onto your picture plots. That's why we see the red and blue here. Okay, if I were to change my gates, okay, you can see that the number of red cells has decreased, for instance. And we've also created a data grid where we can look at those cells one by one and expand the row size and look at those cells in more detail. But we've also gone a step further and created a quantitative analysis in FCS Express with this data. Okay, so the first step in doing that was to create plots for these different populations, or rather these different drug treatments. So let's quickly just go to a two-page view so we can remember how this experiment was designed. Okay, so again, this is the schematic of the experiment. So you can see that different columns correspond to different drugs or different drug concentrations. Okay, and so all of these different dot plots come from different columns. And so let's go ahead and just retitle these dot plots really quick so we can see which column each one comes from. So again, it's very easy to reformat any object in FCS Express. So let's just add here the current gate, because gates were defined using the capability of making well gates in FCS Express. Okay, and I can even make this font a little bit bigger if I want it to stand out a little bit more in my titles. Let's go to a 12-point font. Okay, maybe I want to get rid of that background. Okay, so if I make these a little bigger, you can start to see that they all came from different columns. Okay, so let's go back to our one-page view, and you can space these out, of course, and align them and make the layout look a little more nice. I just want to point out you can even do a landscape view if you'd like. So this is our quantitative analysis. So what we've done here is we've created a regression analysis based on the different data points from each of the columns in our experimental scheme. Okay, so each one of these data points represents a different drug treatment. Okay, and on the y-axis, of course, we have the percent of translocated cells because we're looking at the efficiency of translocation in this data set. Now, this dot plot up here is showing the gates that we defined, and if I change my gate, maybe I want to make my translocated cell gate a little bit more restrictive, hopefully you noticed that my regression analysis and my best fit line that was defined in FCS Express, that's the red line here, automatically updated. Okay? And so the advantage here is you can change your gates on the fly, and you don't have to waste time exporting and re-exporting your data to Excel and then redrawing those plots in Excel or other spreadsheet programs that you might be using. So basically, your quantitative analysis adjusts with you as you adjust your gates. And so that way you can really take a nice interactive look at your data. All right? So this is a very nice figure for publication. We've also gone one step further and created an even more sophisticated report. And so you could easily share this with your colleagues by saving it as a PowerPoint or just even sending them the layout. And I'll talk a little bit more later on about the collaboration options we have in FCS Express. Okay, and so you can see you can customize these histograms to look however you want. Right now we're looking at a stacked histogram view, but if for some reason you wanted to look at a more traditional overlaid view, you could do that as well. Okay, I'm just going to restore that. And here you see we have a 3D view. And I didn't show you how to make overlays today, but I just wanted to mention that it's very easy to make overlays just by dragging and dropping. Okay, so you would just drag one data file into another, or drag one file from your data list into the histogram. Okay, so there's a lot of drag and drop in FCS Express. And if you want to try to drag and drop something, I definitely encourage you to do it because it'll probably work for you. Okay, so let's take a look at one more layout before we start to wrap things up a little bit. And I want to show you this colony counting layout because, you know, colony counting is not something you might think of analyzing in an image cytometry program, but I think it can really save you a lot of time, especially if you're going to be repeating the same types of experiments over and over again, or, you know, even if you're just doing one experiment but there's hundreds of plates and you don't want to waste time counting colonies on 100 plates. So here, this is a very simple TIFF image that was segmented in Cell Profiler, which again is freeware that you can use. And this TIFF image was just taken with a digital camera that was lying around the lab, so nothing too complicated. But it was segmented into Cell Profiler and then imported here into FCS Express. And if let's magnify this picture plot so we can look at it in a little more detail. We've created gates around these two colonies. Okay, so for those of you that are familiar with transfection assays or maybe some sort of other colony screening type assay, you know that you're usually interested in a colony of a particular type. So maybe you're interested in a large colony or colonies of a particular color, for instance, and so you don't want to spend time looking at your pile of 100 plates, but you just want the software to do that for you. So what we've done here is we've created a dot plot based off of this picture plot where we're only looking at the red colonies here. So we've created a gate here on the red colonies because the x-axis here is red. And down here, we're looking at different populations based on size. So we have a large colony gate, medium colony gate, and a small colony gate. Okay, and so here, this picture plot gate has, this picture plot has a gate around this one particular large colony. All right, and so what we can do is, again, look at a data grid, and maybe I want to just drag and drop this gate over here, and I'm only interested in the events that fall in that gate. So this is probably, this large cell here is probably the event you'd be interested in. This large colony here. There's some other objects that have been identified here by Cell Profiler. You can always play with your segmentation routines to make them more or less restrictive if you don't want to identify these little specks as colonies. And if any of you would like any help doing that or establishing a routine in Cell Profiler, just let us know. We'd be happy to talk to you about that so you can then look at your data in FCS Express. Okay? And again, you could add any parameters that you might want to add to this view. So if I want to add eccentricity, okay, so now you see we have another column here for eccentricity. And again, it's really easy to get summary statistics off of your dot plots just by right-clicking and clicking on gate statistics. Okay, so here's a nice gate statistics window, and here's another one here that we formatted a little bit more nicely to be a summary. Are there any questions about what I've talked about so far, about this layout or any of the other layouts? Okay, well, if not, I just want to point out a couple of things that I haven't had a chance to really touch on today. And one of them is compensation. So we do have the ability to compensate data just like we compensate data in flow cytometry. Okay, and you can do that in flow cytometry if you're acquiring multi-fluorescence data. We also have the ability to make bar plots. So I've already shown you in the last layout we looked at a regression analysis with a nice line plot with the regression line overlay. You can also make bar plots if you'd like to with error bars. So there's all sorts of things you can do in FCS Express, and of course we really didn't have a chance to talk about nearly all of them. We also have some great collaboration tools. So if you come up here to the Applications button and come down to Collaborate, you have the ability to publish your layout. And what that will allow you to do is you can create a version of your layout that people who might not have purchased the FCS Express image cytometry program can still look at. And that's accomplished by downloading the free FCS Express reader. Okay, so that's really similar in concept to the Adobe PDF reader, which of course you can download for free and then just look at any PDF document that's online and even make some limited annotations such as highlighting, et cetera, et cetera. So in FCS Express, with the FCS Express reader, which is free, your colleagues can then open up your layout once you've published it, and then play with your gates, maybe examine your summary statistics or add more summary statistics. So it's a really great tool for collaboration. And we also have an on-screen collaboration tool via the internet, which you access just by clicking Start Collaboration. And then you can join a meeting, which is hosted by Novo software, and send that meeting link to your colleagues and just get on the phone with them and just share your screen and look at your data. And you can even share keyboard and mouse control if you prefer. Okay, so let's go back to original layout, and I would just like to take these last few minutes to summarize a few of the things that I showed you so far. So hopefully what I've showed you is that FCS Express Image leverages the most powerful and proven tools of flow cytometry analysis with great new features like data grids and picture plots to improve your image cytometry analysis. And we allow you to move from population-based analysis to single-cell analysis with just the click of a button. New image handling features allow fully interactive analysis between all data plots and your images in cell galleries. And we also combine state-of-the-art visualization, plotting, data analysis, and advanced report generation tools all in one package. So just like I showed you in our batch processing actions, you can send your analysis out to PowerPoint or PDF or Word in just a couple clicks of a button. Okay? And I think most of all, it's an intuitive PowerPoint-like interface really makes the learning curve pretty short for most of you. But if you do need help, we're always really happy to hear from customers. You can email us anytime at support@denovosoftware.com, and we usually get back to you sometimes within a couple of hours. All right, so don't hesitate to send us an email, even if you just need some help getting started with your data. But if you prefer, you can also just access our help from within the program or our website just by coming down here to help. And for instance, you can look at our contents and search for help in our manual by a keyword, or you can click on tutorials. And we have a number of different tutorials which will help you accomplish whatever it is you're trying to do. So here, if you click on, for instance, Introduction to FCS Express for Image Cytometry, that tutorial will help you get started with the program, and it'll go over a lot of the things that we talked about in today's webinar. All right? So before I let you go, I just wanted to show you our website. This is our website, denovosoftware.com. So I really encourage you to download our demo, which is here under Downloads. Click on Demo, and once you click on Download, you can sign up for a free account with Novo software and download a fully functional 30-day evaluation version of the image cytometry program, or even FCS Express Plus, which bundles the flow cytometry program and the image cytometry program into one convenient package. Okay? So if there are no other questions, I just wanted to thank you all very much for joining us today. And I hope that you'll visit us on Facebook and LinkedIn as well and join our users groups where you can get some really handy tips for analyzing your data in FCS Express Image Cytometry. Thanks so much. Have a great afternoon.